Good morning, good afternoon, and good evening, everyone. Welcome to our global webinar series hosted by ELab Science Bioinnovation. We are thrilled to have participants from around the world joining us today for this advanced session on ELISA. I am Dr. Ikra Hafiz and I will be your host as we explore the end-to-end approach of ELISA from experimental design to precise detection and ultimately to impactful scientific delivery. Over the next hour, we will learn how ELISA technologies enable accurate biomarker measurement, support breakthrough discoveries, and accelerate advancement across immunology, disease research, and therapeutic development. Let's begin this journey from branch to breakthrough. Okay. Okay. Before we jump into today's topic, let me give you a quick introduction to ELab Science. We started back in 2009 and since then we have grown into a global life science company. With a presence in over 150 countries and multiple international certification like ISO 9001, ISO 13485 and CE. Our products have been cited in well-known journals like Nature, Cell and Science. We began with basic bioresearch reagents and ELISA kits in 2011, and over the years we have expanded to include cell culture solution, CLIA kits, antibodies, proteins, IHC, Western blot kits, metabolism assays, food safety, animal disease kits, and flow cytometric antibodies. More recently, we have added advanced tools like induced differentiation media, exosome isolation kits, ELISpot, Cellacont, primary cell isolation kits, and immune cell isolation kits. Along the way, we have been recognized with awards like Sites Supply to Watch and the Kotler Award for Customer Value. All of these reflect our commitment to innovation and high quality research tools, empowering scientists to design with precision, detect with confidence, and deliver breakthroughs. A few housekeeping notes are here. First, to keep the momentum, and please keep your microphone muted during the presentation. Submit any question via Q&A panels. We address them at the end. For other issues, please write in the chat box. A digital participation certificate and the session recording will be sent to all registrants. And attendees also have the chance to receive free samples of selected ELISA Science products. Highlights are given in the slides and the full list with eligibility and claim instruction will be sent to your registration email. Now it's my pleasure to introduce today's speaker, Dr. Sundar Qureshi, who will guide us through the journey of ELISA from assay design to accurate detection and impactful scientific delivery. As we explore the theme design, detect, deliver, ELISA from bench to breakthrough, we will see how robust assay development and reliable detection platform enable researchers to generate high quality data and accelerate discoveries across a wide range of the science applications. Dr. Qureshi serves as a global scientific liaison at ELAB Science Bioinnovation. She operates at the forefront of life science innovation, connecting scientific strategy with real-world application to transform complex technology into practical and high-impact solutions. She works closely with researchers and partners worldwide across immunology, cell biology, protein analysis, and advanced assay platforms, including ELISA, while leading numerous international training and scientific engagement that drives stronger decision, smarter, smarter adoption, and measurable results. With a strong global footprint in scientific communication and technology. Dr. Qureshi brings clarity, direction, and momentum to modern life science researches. We are excited to learn from her insights today. Dr. Sundars, it's up to you.
First of all, I would like to welcome everyone. Good morning and assalamu alaikum. ELISA has been a cornerstone of bioscience. Yet it also value, its core value lies in how it connects the basic discoveries to the translational outcomes. Today, we'll explore how rigorous design and smart kit selection produces reliable data, how integration with complementary assays stands out and expands the scientific insight, and how innovation in ELISA are driving translational research from uncovering mechanisms into the lab to performing the preclinical studies and real-world applications. By the end, I hope you will see how ELISA delivers not just data, but pathways to discover, validation, and breakthrough impact. So let's begin. My today's presentation will be divided into three sections. We'll start with designing a strong ELISA experiment, then look at how to select the right kits and avoid the common errors. Next, we'll explore how ELISA can be integrated into other assays for deeper insights. And finally, I will discuss its role in translational research and some latest innovations shaping the field. If you have any questions, I would like to ask you to write in the Q&A section, and we will address them one by one at the end after my presentation. Okay, so in this first section, we'll focus on how to turn ELISA question into a properly designed ELISA experiment. I'll guide you through the essential steps to make sure that your study is reliable, reproducible, and suitable for publication. Okay, let's start with a quick refresher because I believe there are some students over here. So I'm just giving a brief introduction on ELISA. ELISA, or enzyme-linked immunosorbent assay, is a method to measure specific proteins in the sample. It works because antibodies can recognize and then bind to specific proteins. These proteins are mostly the antigens. In the typical ELISA, the first antibody is attached to the surface and then captures the target protein from the sample. So here you can see in this picture, this is our capture antibody, which has been plated or coated on the ELISA plate. Then we have a second antibody, which is our detection antibody. This detection antibody is linked with an enzyme, which binds ultimately to our target protein in the or our analyte or the biomarker which we are detecting. So when the substrate is added, and what happens, it produces the color or some signal change. So the strength of the signal shows how much protein is present in the sample. This method is very sensitive and widely used in research, diagnostics, and pharmaceutical studies. Even if you are new to it, once you follow these basic steps, you can measure the proteins accurately and reliably. Every ELISA experiment starts with a clear research question. Firstly, decide exactly what protein or what molecule you want to measure and why it matters. For example, if you want to measure the level of a cytokine like IL-6 in the blood samples to study inflammation, or a growth factor in cell culture to see how cells respond to the treatment. Next, you have to think about your hypothesis. What relationship or outcome do you want or do you expect? For instance, you might hypothesize that treated cells will produce more of a specific protein than the untreated cells. Once that is cleared, you are able to design the ELISA experiment. This includes planning and experimental setup, choosing the right controls, and selecting a detection method. You also need to consider the type of sample, which is serum, plasma, or cell culture supernatant, any kind of sample which you have. Then one most important thing is the appropriate dilution. I will go into detail of all of these one by one, but just let's focus on the key things, which are the sample type, dilution ratio, and what is your specific question. Finally, we have to consider the expected results. What measurements will support or challenge your hypothesis? Adding these examples make it easier to visualize and ensure that your data is meaningful and reliable. Okay, now the first part, a strong research question. A strong research question needs to be precise and measurable. Vague questions like what proteins are involved in the inflammation or does the treatment affect the cells are too broad and make it hard to plan the experiments. Well-framed questions, on the other hand, clearly state what will be measured in which samples and under what conditions. For example, instead of just asking which proteins are involved, you could ask which cytokines are elevated in plasma of the patient with X condition compared to healthy controls. Or instead of asking if the treatment affects the cells, you could ask, does the drug Y reduces IL-6 secretion in THP cells after 24 hours of treatment? So here, the questions are quite clear, systematic. Another example is rather than asking why some samples react differently, you could ask how does sample A compare to sample B in TNF alpha levels measured by ELISA? So framing your question this way makes it much easier to choose the right assay, set proper controls and analyze your data so that your results are clear and reliable. Okay, now not all ELISA formats work the same way. So choosing the right one is important. There are several different methods like direct ELISA. This is simple and fast, which makes it good for measuring the proteins that are present in high amounts like albumin in blood. However, it is less sensitive and has limited flexibility. In indirect ELISA, we add a secondary antibody or second antibody, which amplifies the signal. This amplification ultimately improves the detection and allow more versatility, but it does take an extra step. On the other hand, we have a sandwich ELISA, which is both highly sensitive and highly specific, making it ideal for detecting low abundance proteins in the biomarkers, such as cytokines in patient plasma samples. Competitive ELISA works differently. It is used for smaller molecules like hormones or drugs, where the signals decreases as the target concentration increases. So when we are looking at the standard curve of competitive ELISA and the sandwich ELISA. In sandwich ELISA, the standard curve will move in the direction. So it is the directly proportional to the concentration. However, in competitive ELISA, the standard curve is always inversely proportional to the concentration. By understanding the strengths and limitations of each format, you can pick the approach that gives most reliable and accurate results for your particular experiment. This way, you can choose the ELISA format that fits your sample, your research goal, and then ultimately get the results for the proteins or molecules you are measuring. Now, the controls are also essential for getting the reliable ELISA result. First of all, the positive control, they show that assay is working properly, and while the negative controls reveal any non-specific signals. Blank will measure the background noise, so you know that which part of the signal from the assay itself. Including these controls make it much easier to spot the errors early and avoid drawing the wrong conclusions. That is why it is important to plan your controls before you start the experiment rather than adding them later as an afterthought. Now, controls are one part of the equation. The other part is the replicates, which are key to making ELISA results reliable. Biological replicates come from different people, animals, or samples, so they show real differences between individuals or conditions. However, the technical replicates are repeated measurements of the same sample, usually in multiple wells. Like here, you can see we have added sample 1 and sample 2 two times. The sample is same, but the replicates are in duplicate. So they help catch any variation that comes from the experiment itself, like tiny differences in pipetting, how the plate is handled, or slight changes in the detection, or sometimes any temperature fluctuation in the incubator. So by using both the biological and technical replicates, we can tell what is the real effect and what's just experimental noise. So doing this properly also makes your data stronger and your conclusions more trust and publication worthy. How you handle and store your samples really affect your ELISA result. Proteins in the blood, plasma, cell culture, or tissue samples can start breaking down if they are left at a room temperature for too long, or if they go through repeated freeze-thaw cycles. That's why it is important to freeze the samples quickly and minimize the number of times they have been thawed and or refrozen. Spinning the samples in the centrifuge removes the debris that could interfere with assay or give you misleading results. There are some clear labeling, which is also very, very essential. You don't want to mix up the samples, right? Or waste time figuring out, okay, which tube is which? Let me see. So we don't want that. So for long-term storage, we have also have to keep the samples at the recommended temperature, like minus 80 degrees. And when it's time to use, thaw them slowly on ice or in the 4-degree refrigerator so that the proteins stay intact. Using fresh reagents and storing them properly is equally important, because even small changes can make the results change entirely. I know some of you might be thinking, who doesn't already know this? But trust me, we all make such mistakes, and even small errors in the sample handling can really affect your results. Paying attention to these tedious details makes you save your time and makes your measurements much more reliable, and you will also save a lot of frustration later as well. Okay, getting the right dilution is also key to good ELISA results. If your sample is too concentrated, the signal can max out and you won't be able to measure it accurately. If it's too diluted, you may not detect anything at all. One common approach is serial dilution, where you dilute the sample step by step, usually by the same ratio each time. For example, you could take 1 part sample, mix it with 9 parts of buffer to make 1/100 dilution, so that and so on. This lets you see how the signal changes at each step and find the range that works best for your assay. You can also follow the ratios suggested in the literature for your sample type. Running a small pilot experiment at the start also helps you pick the best dilution before doing the full study, so your measurements are accurate and meaningful. If you are even unsure after that, eLab Science Tech Support is always there to guide you for your dilution for your particular samples, and we will help you in selecting the right dilution. Now, getting a good ELISA data isn't just about running the assay, it's also about documenting it properly. Journals expect you to clearly report the number of samples. So, for example, showing the data from 20 samples rather than just 3 gives your results strength. They also want to know the controls you use, like positive controls, negative controls, blank controls, and the replicates, such as running the samples in duplicate or triplicate. Explain how you have analyzed the data and calculated things like average, standard deviation, or relative concentrations. Assay validation is important too. You might show that your ELISA can reliably detect IL-6 in plasma at the expected range just by showing the validation. And any changes you made in the standard protocol as well, like adjusting the incubation time, should be noted. So following these steps makes your results reproducible, credible, and ready for peer-reviewed publication. Now, before moving on to the next section, it is very, very important to talk about why sometimes ELISA results just don't look right. And it is really, really frustrated sometimes. And usually the reasons fall into three categories: the kit, your sample, and how the experiment is run. Starting with the kit, if it's been stored incorrectly, contaminated, or if you accidentally mixed the reagents from different kits, the results can go off. The fix is simple. Keep everything at the recommended temperature. Don't mix the components and let the kit reach room temperature before you start. Equilibrate everything. Next, the samples. Problem here include poor clarification. The samples might not be clarified before starting the experiment. Then bacterial contamination in the sample can also cause these problems: hemolysis, hypothermia, or repeated freeze-thaw cycles. All of these problems would ultimately lead to the abnormal ELISA results. Even something as small as incorrect dilution can throw the whole assay away. Make sure that the samples are properly prepared, stored, and diluted exactly as the protocol says. Finally, the experiment itself. Incubation temperature and time are critical. We know that. Pipetting has to be precise as well. Avoid any kind of bubbles in the wells and be careful with the biotinylated antibodies or HRP conjugates because they can also ruin your experiment. Adding them in the wrong order or wrong concentration can entirely ruin your results. Standardized washing steps make sure that the termination step or stop solution has been complete. And check that your plate reader is set to the right wavelength, usually 450 nanometers. So looking at all these three areas, kit, sample, and operation covers most of your common causes of abnormal results. And if you follow these checks, you can usually pinpoint the problem and fix it quickly. And I am sure that after this, you won't be able to get your abnormal results. And now wrapping up this section, strong ELISA results start with asking the right question and planning carefully. So every choice from assay format to sample handling shapes the reliability of your results. Good planning today makes meaningful, trustworthy data tomorrow. Okay, now let's move to the next section. In this section, we'll look at how to choose the right ELISA kits for your research needs and how to combine ELISA with other assays to get a fuller picture of your samples. The goal is to work smarter, not harder, and make your experiments more informative and reliable. Okay, there are many different types of ELISA-based kits available in the market. And eLab Science also offers a wide range covering different needs, from traditional ELISA kits to high sensitivity, mini sample formats, multiplexing systems like APTplex, and even ELISA spot and cell-based assays. But the key question is not how many options are available. It is how to choose the right one for your experiment. Start with your sample type. Are you working with serum, plasma, cell culture supernatant, or cell lysate? For cell-based samples, CellQuant kits are designed to measure proteins directly from the cells. If your sample volume is limited, Mini sample kit is only the one which requires a very small sample volume. Then think about what exactly you are detecting. Traditional ELISA are good for measuring one protein at a time. High sensitivity kits are used when your target protein is present at a very low level. Then we have the viral protein kits that are specifically designed to detect the viral antigens or antibodies. If you want more flexibility and lower cost, uncoated ELISA kits lets you coat the plate yourself. Then we have the clear kits, which uses the chemiluminescence instead of color change, which gives higher sensitivity and a wider detection range. Next, we have to consider how much information you need. If you want to measure multiple cytokines or biomarkers in one sample, APT-plex allows multiplex detection instead of running many separate ELISAs. If you are interested in cell function, like how many cells are secreting a cytokine, ELISpot gives results at a single cell level. So also think about the workflow as well. If you need faster results, Quickie Pro kits shorten the assay time to up to just 90 minutes. And for the applications like food safety or environmental testing, there are specific kits for drug residues, mycotoxins, and vitamins. So instead of just picking a kit based on availability, match the kit to your sample, your target, and what you want to learn from the experiment. That's what helps you get clear and reliable data, publication ready results. I have seen many customers that they only know, okay, this is our target. Tell me which kit I can use. This is not how a perfect ELISA data works. You have to be very, very clear about what you want to do with your target. If you only tell us, okay, I want to detect this target, any kit could work. But do you think that is suitable for your experiment? No, we can't guarantee that. This is what your experimental question will tell you. If you can create your hypothesis, your question clearly, then you can clarify which ELISA kit suits you best. Okay, choosing the right ELISA kit is not just a purchasing step, as I just mentioned, that some people just ask, okay, I want the kit for TNF alpha. Give me the TNF alpha. There are so many TNF alpha kits available. It directly affects the quality of the data. So a well-validated kit gives us the results that are accurate and reproducible, so you can trust what you are seeing. On the other hand, if the kit is not suitable, it can introduce errors that are very hard to fix later, no matter how much troubleshooting you do. Good quality kits also make a bigger difference, and when it comes to publishing as well, because reviewers look for reliable data, and using a validated kit adds confidence to your results. In many cases, ELISA data is used to guide further experiments or even support the clinical decisions. So the impact goes beyond just one experiment. You might be saving a life, or you might be killing someone. So it is important to treat the kit selection as a part of your experimental design, not just something you order at the beginning from some shopping app. Okay, when we are selecting ELISA kit, specificity and sensitivity are very, very important. ELISA kits are usually designed for a specific species. So a human kit is meant to detect human proteins, a mouse kit for mouse proteins, and so on. If you use the wrong one, the antibody may not bind properly, or it may bind to something else and give you misleading results. This becomes especially important in experiments involving multi-species like xenograft or co-culture studies. That's why it is important to check the data sheet and confirm the species compatibility. Some kits also provide cross-reactivity data, which helps you see whether the antibody might bind to similar proteins from other species. Then sensitivity is another key factor. It tells you the lowest amount of protein the kit can detect. The dynamic range shows the concentration range where the results are accurate. You will often see terms like LOD, which is limit of detection, and LOQ, which is limit of quantification. These help you decide whether your target protein falls within a measurable range. And then we have the last thing, accuracy, which tells you how close your result is to the actual value. So looking at these parameters before choosing the kits help you avoid unreliable data and saves time later. When you look at a kit's data sheet, the validation data is one of the most important part. For example, recovery tells you whether the kit accurately measures your target, like 95% recovery rate shows within the acceptable range of 90 to 110%. Linearity shows whether the assay responds proportionally across concentration. An R square value of 0.99 indicates very good correlation between the expected and observed values. Precision is also the key. Intra-assay precision, which looks at the variability between replicates in the same run, might be 6%, showing low variation. Then the intra-assay precision measuring the consistency across multiple runs might be 9%, which is still very good. If any of these values are missing or unclear, that's a warning sign. Kits from manufacturers that provide clear, complete validation data gives you much more confidence in your results. So paying attention to these metrics upfront help avoid any unreliable data, saves you a lot of time, a lot of frustration later. Okay, when you are working with ELISA, there are two main types of pitfalls or mistakes in the kit selection or errors during the execution. For the kit selection, common mistakes include choosing the wrong species. For example, using a human kit on the mouse sample or picking a kit with insufficient sensitivity or wrong dynamic range for your target protein. Another trap is ignoring the validation data. Without checking the recovery, without checking the precision or linearity, you might end up with unreliable results. Execution errors happen when even with the perfect kit. Pipetting mistakes like inconsistent volume across the wells, you can literally skew your data. Then inadequate washing may leave unbound antibodies that create high background. You will ruin your data. Incorrect incubation time, whether too short or too long, can also affect the signal strength. Even a high quality kit can't fix these issues. So being aware of these pitfalls ahead of time lets you plan properly, like double checking your sample type, validating the kit's sensitivity, and practicing careful pipetting. You can also save the samples, reagents, and the data which you can trust. ELISA gives you valuable information about protein levels, but it only shows one piece of the puzzle. To get a fuller picture, it's often useful to combine it with other assays. For example, qPCR that can show the gene expression behind the protein. Western blot can confirm the protein size and specificity. And flow cytometry can tell you which type of cells are producing it. Immunohistochemistry detects the antigen in the tissue sample. CBA allows the users to quantify multiple proteins simultaneously. This is the multiplexing technique. So by integrating these methods, you can cross-check your results and add the biological context. It also helps prevent mistakes that can happen if you rely on a single technique. Using multiple approaches together makes your conclusion stronger, more reliable, and more convincing to the publication. ELISA measures the actual protein levels in the sample. This is what is universal, everybody knows that. While the qPCR looks at gene expression. Why we need to use both of them? Using both of them together helps us link what's happening at messenger RNA level to the proteins that are actually being produced. For example, in one study, increases in the cytokine genes like IL-1 beta, IL-6, TNF alpha, or IL-10 in the kidney tissue were matched by higher cytokine levels in the serum. You can see here the data is truly similar. So what is this showing? This shows that gene changes were reflected in the real protein output. At the same time, combining these assays can reveal interesting differences as well. Sometimes messenger RNA goes up, but protein levels doesn't change, which tells you more about the regulation in the system. So doing both ELISA and qPCR gives more complete picture and makes your conclusions much stronger. ELISA also can, if we combine ELISA with Western blot, we know that ELISA will tell us how much protein is present, but it doesn't confirm the protein size or whether the signal is coming from the right protein or not. That's where Western blot comes in. For example, in this particular study, ELISA showed that MUC5AC protein levels increased in primary human nasal epithelial cells after the treatment in IL-19. Western blot then confirmed that the signal was actually this protein, showing the correct protein size. Here you can see it shows the correct protein size, and the data is now reliable. Using ELISA and Western blot together helps us measure the protein quantity and also verify the specificity. ELISA is high throughput and gives you broad measurement, while Western blot makes sure that these measurements are accurate. So combining these two reduces the false positives and gives much more confidence in the results. Okay, we know ELISA measures how much of the protein, like a cytokine, is secreted into the cell culture supernatant, giving you overall quantity, but it doesn't tell you which cells are producing it. That's where flow cytometry comes in. Flow cytometry can identify the specific cell types making cytokines. For example, in COVID-19 studies, TSNE maps for the flow cytometry shows that immune cell produces particular cytokines, while the ELISA measures the total cytokine levels before and after treatments like lyophilization. So by combining the ELISA and flow cytometry, we connect the amount of protein to its cellular source, to its cellular core. This gives a much more complete picture of immune responses, showing both the magnitude of the protein secretion and which cells are responsible. So to wrap up this section, what we learned that choosing the right ELISA kit and running it carefully is the key. You can also combine ELISA with other assays to get a fuller picture of your results. For example, CLIA is also very sensitive and fast. So pairing it with ELISA helps when the protein levels are very low. So you need the quick results, combine CLIA detection with ELISA detection. Immunohistochemistry also shows not just how much protein is there, but where it is located in the tissue, giving a special context to your ELISA measurements. Multiplex bead arrays lets you measure the several proteins at once from the same sample, saving time and giving a broader view as well. And then we have the CBA, cytometric bead arrays, which work similarly. So measuring the multiple cytokines in one go, while ELISA helps verify the results. Using these combinations make your data stronger, more reliable, and more easier to interpret. Now, in this final section, we are going to see how ELISA helps take discoveries from lab and apply them in real-world translational research. We'll also touch on some of the latest trends and innovations that are making ELISA even more powerful and expanding the ways it can be used in modern research. So let's talk about ELISA, how ELISA actually moves the research from lab to real patients. In lab, we might use ELISA to measure inflammatory proteins like IL-6 or TNF alpha to see what's driving a disease process, or look at VEGF if we are studying the tumor growth. Then, when we test a new drug on preclinical models, ELISA can show whether the treatment is really having an effect. For example, whether IL-6 level drop in mouse model of arthritis or if CRP decreases in an inflammation study. Moving to the clinical side, ELISA is still the same tool, but now it helps monitor patients. You might track the antibody responses after COVID-19 vaccine, measure the troponin levels in heart disease, or follow the cytokine levels in autoimmune patients. What's amazing is that the same assay works across all these steps. Lab experiments, animal studies, and patient monitoring give us continuous reliable data. That's why ELISA is such a cornerstone in translational research. It doesn't just give us numbers, it tells a story of how discovery in the lab can actually make a difference in real human health. Now, take an ischemic stroke, for example. When tissue gets damaged, danger signals activate NF-K beta and MAPK pathways, which then trigger the NLRP1 and NLRP3 inflammasomes. That leads to the release of cytokines like IL-1 beta and IL-18. These cytokines act on neurons, glial cells, or immune cells, ramping up the inflammation, while caspase 1 pushes the cells towards the apoptosis. With ELISA, we can measure IL-1 beta and IL-18 in the tissues or blood, giving a clear picture of how active these pathways are. Researchers can even test the potential therapies. If a drug inhibits NF-K beta or MAPK, you will see IL-1 beta and IL-18 level drops, and cell death decreases. So ELISA isn't just measuring numbers, it's showing us exactly how these signaling pathways drive the disease and where we might intervene. In the preclinical research, one of the most powerful things ELISA does is help us track the biomarkers. For instance, you might take IL-6 in serum or plasma, see how severe the inflammation is, or TNF alpha in the cell culture supernatant to predict how an immune response might react to the treatment. Then MMP9 can tell you about tissue remodeling in stroke or cancer. VEGF gives you insights into angiogenesis and the tumor growth, and CRP is a general marker for inflammation or cardiovascular risk. ELISA makes it possible to measure these proteins accurately and reproducibility, even in the tiny amounts. But by doing this, we can figure out which molecules really matter, which helps when deciding how to design clinical trials or choose the patient who might benefit most from the therapy. Essentially, ELISA bridges the gap between lab studies and real-world clinical applications by giving us solid, actionable data on biomarkers. This technique has been the workhorse of drug development for decades because it lets us track the proteins and see if the drug is really making an impact. For example, in the metabolic disease research, we might measure adiponectin or leptin to see how treatments affect the metabolism. In cardiac studies, NT-proBNP or troponin levels reveal how the heart responds to the new therapies. If we look at immune modulation, for example, IL-10, TGF-beta, or granzyme V, they can show whether the immune system is being activated or suppressed. Even in the infectious disease studies, you could track viral proteins like influenza, RSV, F protein, or monitor the antiviral effects. Or even there are some other viral proteins like SARS-CoV-2, N protein, or HIV P24. They can be quantified to assess the treatment effect. During the pharmacodynamic studies, ELISA measured these proteins over time, showing how drug affects the system and helping optimize dosing schedules as well. In early screening, it quickly flags when the compounds are active and worth pushing. So the beauty of ELISA is that it's fast, sensitive, and reproducible. So you can use it at multiple stages, from initial screening to preclinical studies and translational applications. It really turns protein measurement into actionable insight that guide the drug development. Multiplex ELISA is a real game changer when you want to get more information from less sample. Instead of measuring one protein at a time, like in traditional ELISA, multiplex platforms let you measure multiple proteins all at once. This saves precious sample volume and cuts down on experimental time, ultimately cutting down a lot of cost. It especially also handy in immunology or biomarker studies where you want to look at whole panel of cytokines or signaling proteins together. For example, you could measure IL-1 beta, IL-8, and IFN gamma in the same run instead of running 3 separate assays. Multiplex ELISA boosts the throughput, makes the experiments more efficient, and still gives reliable, high-quality data. Sometimes your target protein is just too rare to detect with a standard ELISA. That's where high sensitivity methods come in. Ultra-sensitive ELISA can pick up proteins in picogram per ml range, perfect for early biomarkers or proteins expressed at very low levels. Single molecule detection makes it even further down to the femtogram level, letting you measure proteins that are typically invisible in regular assays. These techniques also need very little sample, less than the standard ELISA, and use enhanced detection methods like chemiluminescence or fluorescence with the amplification to boost the signal. So if you're working with the rare proteins or pushing the limits of diagnostics, these high sensitivity approaches open up possibilities that were impossible before. Now move on to the omics. Omics is where big pictures come alive. Transcriptomics like RNA sequencing or qPCR. Let us see which genes are being turned on or off in a cell or tissue. Metabolomic tracks small molecule and metabolic pathways, so we know what's actually happening chemically inside the cell. Proteomics pathways helping us understand the machinery behind the biology. Now, ELISA fits in perfectly here. It measures the actual protein levels that the omic data predict. For example, if RNA sequencing shows the inflammasome genes like NLRP3 are active, ELISA can confirm whether cytokines like IL-18 or IL-1 beta are really being produced. System biology then ties all this together, letting us model networks and see how proteins, genes, and some other metabolites interact in the real biological system. Putting all these pieces side by side turns scattered data into a clear picture. You can actually see which proteins, genes, or metabolites are driving the process, and it points straight to the meaningful biomarker or potential drug targets. That's what makes the research more reliable, more closer to the real world impact. Okay, as we finish, it's interesting to look back at how ELISA has evolved. Starting from 1960s with radioimmunoassays, researchers sought ways to measure proteins more safely and reliably. In 1971, Parliament and they introduced the first enzyme-linked assay, replacing the radioactive labels with enzymes. Uh, Through the 70s and 80s, ELISA moved into diagnostic and research labs with sandwich and competitive formats, becoming standard. Automation in the 90s and then miniaturized high sensitivity ELISA kits in 2000 made the experiment faster and more precise. In recent years, multiplex and single molecule platforms have allowed measurement of multiple proteins at very low concentrations, integrating with other techniques to answer complex biological questions. After all these decades, it's simple, reproducible, and fast, making it a go for both the lab experiments and translational research. ELISA is still reliable. Its history shows that solid, dependable methods can keep giving value, even the technology move forward. So it's not just a technique, it's a legacy built to last. That's all for today's presentation. If you have any more questions, I have mentioned my email ID in the first slide, and you can also write to me at events@elabscience.com. I will always be ready to help you. And now I will give the floor back to Miss Dr. Ikra, and I will definitely look through the messages in the chat box. So you have a lot of questions here in the Q&A session as well. So let's try to give the answers clearly, but we have very, very short time right now. Okay, Ikra, I think you can just quickly wrap up.
I will start again. Thank you, Dr. Sundas, for that insightful session on how ELISA supports high quality research from design to data generation. To further your research work in this area, I would like to introduce ELAB Science comprehensive ELISA product portfolio designed to streamline every step from assay development to reliable detection and final data delivery. Our ELISA kits from the core of these portfolios offering a wide range of formats tailored to different research needs, including traditional ELISA, rapid workflows, and high sensitivity solutions, ensuring flexibility without compromising accuracy. For researchers looking to build or customize their assay, we also provide key components such as uncoated kit and matched antibody pairs, enabling greater control over assay design and optimization. In addition, our advanced platform supports multiplexing and expanded analytical capabilities, allowing you to detect multiple targets efficiently within a single experiment. Beyond kits, we offer a full range of spotting reagents and auxiliary products that help maintain consistency, improve reproducibility, and simplify your workflow from start to finish. These solutions are optimized to help you design with confidence and detect with precision and deliver meaningful results. As we know, accuracy biomarker measurement starts with choosing the right detection tools. ELAB Science provides a wide range of ELISA kits designed to help you measure key targets across different research areas with confidence. You can start by selecting from a broad panel of validated biomarker covering inflammation, oxidative stress, metabolism, neurobiology, and cardiovascular research. These kits are optimized for sensitivity and specificity, allowing you to detect even low abundance target with consistent performance. Each assay kit is designed with a straightforward workflow, making it easy to move from sample preparation to data generation without unnecessary complexity. This means you can spend less time troubleshooting and more time focusing on interpreting your results. The flexibility of these kits also allows you to adapt them to different sample types and experimental needs. Whether you are working with serum, plasma, or cell culture samples. Overall, these ELISA solutions support a smooth transition from experimental design to accurate detection and reliable data delivery, helping you to generate results that truly support your research goals. As a reminder, webinar attendees are eligible to request one free sample from this given product list, and full detail about the claim process will be sent to you by your registration email. So now we will move on the question and answer segment. Please type your question in the Q&A panels and we will address as many as possible. If we are unable to answer all questions during this time, you can send them to events@elabscience.com and our team will provide detailed responses. So I have got a first question. The first question is, how do I choose between sandwich, competitive, and indirect ELISA from my protein? Dr. Sundar, can you answer?
Okay, yeah, sure. Actually, the question is quite simple. How do you choose between the sandwich, competitive, and indirect? I think I mentioned in the beginning, sandwich ELISA is actually ideal for the larger proteins with multiple epitopes because it uses two antibodies for higher specificity and sensitivity. Competitive ELISA work best for the small molecules of proteins with a single epitope. Here, the sample competes with the labeled antigen for binding. Indirect ELISA is usually for detecting the antibodies in sample rather than the antigen, making it suitable for serology studies. So your choice depends on the molecular size, number of epitopes, and whether you are detecting the antigen or the antibody.
Okay, the second question is, my standard curve is not linear at higher concentration. So what should I do? Dr. Sundar, can you answer this question?
Okay. Okay, I think this question could wait, as someone wrote here that we ordered for some ELISA kit, but they did not specify the detection range. Mr. Ibrahim, can you please write us at events at elabscience.com if you can hear me properly? So that when you will send us the exact ELISA catalog, we will check in our backend and we will tell you the detection range. Basically, the detection range is mentioned on the ELISA kit, but if it's not mentioned, please do ask us before starting your experiment. Okay, now for the question which Ikra has just mentioned, the person asked, my standard curve isn't linear at higher concentrations, what should I do? Non-linear behavior at high concentration usually indicates the saturation. So your sample may exceed the dynamic range of the assay. You have to dilute your sample and then rerun it. And make sure that the standards are freshly prepared and properly mixed. Also check the incubation times. Sometimes over incubation can also exaggerate the saturation.
So here is another question. Why do I see signal in my blank or negative control wells?
Actually, this often results from non-specific binding or contamination that you are seeing signals in your blank well or even the negative controlled wells. It is definitely due to non-specific bindings, which can happen due to contamination. It could also be due to inadequate washing, because sometimes people don't wash properly. cross contamination of reagents can also cause this, or using improperly stored buffers. That is also a main cause. So you have to ensure that washing steps are thorough. Avoid touching the plate bottom with the tips, because whenever you are adding your standard or sample, make sure that your pipette tip is not touching the wall, not touching the base of the plate, because that can also contaminate the reagents. And another thing, you must also use the fresh reagents, freshly prepared reagents.
So here is another question, and I think it's very important. The person asking, my replicates are inconsistent. So what's going wrong with this experiment?
Variability can arise from pipetting errors as well. Bubbles in the wells, inconsistent incubation temperatures, or even uneven washing. So you have to use the calibrated pipettes, ensure the consistent technique, and make sure that the plate level is equilibrate during the incubation. Always avoid touching the bottom of the wells when you are washing as well.
The last question is, uh, the person is asking, is it okay to skip washing steps or shorten incubation time to save time during the experiment?
No, definitely not. Skipping washes or shortening the incubation compromises the accuracy and reproducibility. Washing removes any unbound material and reduces the background. Incubation ensures sufficient binding. So, cutting the corners often causes false positives, high background, or even sometimes inconsistent results. So, definitely, it's a big no. You cannot skip the washing steps. You cannot shorten the incubation time just to save the time. It is very, very important to stick to the protocol.
Yes.
Okay, there is another question. Already the sample concentration is too low. Does the dilution helps or not? If the sample, the concentration of your analyte is already low in the sample, um, basically you have to be sure that you are using the right type of kit. If the concentration is already low, you can go for high sensitivity ELISA kits rather than the traditional ELISA. It will boost up the signal, and you will be able to detect your sample, and the concentration or the dilution ratio will be according to the high sensitivity ELISA kit.
So I would like to say thank you all for joining us and engaging with this session. And we hope Dr. Sundar's presentation, along with the overview of eLife Science ELISA solution, provided valuable insight into improving assay performance, ensuring data consistency, and supporting confident decision making in your research. If we were unable to address any of your questions during this session, please feel free to send them to events@elabscience.com, and our team will provide detailed responses after this webinar. Here, I would like to remind you, don't forget to check your registration email for details on claiming your free sample and accessing this session recording. And we look forward to seeing you in our future webinars. Have a great day and happy experimenting.
Bye bye, everyone.
Take care. Bye bye.